And Ashlee, you have your presentation. You will run it on your own. Is that how we're doing this?
That works for me.
Okay, great. And you know how to share it. Sometimes there's a little bit of confusion with what is the presenter's view and then what is the full screen view for the viewers. And you might need to do a little switching around, but no worries, we have some time.
Yeah.
Since it's 10 a.m. and I see that we have quite a few participants already, I want us to get started, and I want to thank you all for joining us today for the COVID-19 Research Spotlight Series. My name is Tommy Soers. I'm the Assistant Director of the Purdue Institute of Inflammation, Immunology and Infectious Disease. And it is my pleasure today of welcoming Ashlee Colbert from Jacqueline Linnes' group in BME. Ashlee has been working on rapid SARS-CoV-2 point of care detection technologies, and we're really excited to hear the type of progress that she's been making. I'm also very excited to welcome Caroline Plescia from Rob Stahelin's group in MCMP. Caroline is modeling SARS-CoV-2 entry and egress with virus-like particles. We're very excited to welcome them both. The way this is going to work is that Ashlee is going to start. She's going to give her presentation. It's going to take about 20-25 minutes, and then we will open it up for questions. If you would like to ask questions as Ashlee's going along, please use the chat box. And if you look on your screen, you should be able to open the chat box as an option from the icons that open up on your WebEx graphical user interface. So on the chat box, if you want, as we're going along, so that it's not disruptive, please put your questions there. And then after Ashlee's done presenting, I can take those questions up and we can have Ashlee field them. At that point in time, if there is somebody who would like to say something, then you can unmute your microphone. But what I will ask is that everybody mute their microphones during the presentation so that Ashlee can kind of move through the presentation, and then we can open it up for questions if you'd like. We need to be mindful also because Caroline needs to also do her presentation for the next 20-25 minutes, and then we open it up for questions at the end there too. So we want to allow for enough time. If you're having trouble with the bandwidth, I recommend that maybe you shut down your video. We'd love to see you. I'd love to see your faces, but it might improve your bandwidth if you're having some trouble hearing or some trouble looking at seeing some of the presentation details. So with that, it is my pleasure of welcoming Ashlee Colbert. Take it away, Ashlee. I'm going to end sharing my presentation and I'm going to pass it on to you.
Okay. Just want to remake sure you guys can hear me, right?
Yes, we can. Fantastic.
I was trying to put on my headphones and it just went crazy. So I guess we'll keep it.
I know what you mean.
Share.
Perfect. And we can now see you sharing your screen.
Oh, we're sharing Microsoft. Okay, good.
Okay, perfect.
All right.
Okay, ready to go.
Yes. Excited. Okay, so uh, again, I am Ashlee Colbert. I'm a PhD candidate in biomedical engineering, and I'm actually co-advised by Dr. Jacqueline Linnes and Tamara Kinzer-Ursum. And so I work on a lot of point of care detection with the smartphone device that I'll be talking about. So just kind of an outline, we're going to go over the significance and need for this point of care type of detection. We'll look at the current diagnostics for COVID-19, and then I'll introduce you to the method we use, which is a mouthful. It's partial diffusometry paired with loop-mediated isothermal amplification, or PD-LAMP. And then we'll go through some of the results that we've gotten so far for detecting SARS-CoV-2. So global health is all around us. This is really just showing all the different networks between air and shipping routes. And in the past 20 years, there's been 5 pandemics, including COVID-19. And so it's really, really important to begin to detect rapidly. And so these numbers here are for COVID-19, and this was yesterday, so they're probably, unfortunately, a little bit more increased today. Actually, when I had them last week, I think it was like 8 point something million, and now it's at 11. So that is really crazy on how many cases there have been. And what's really alarming is the amount of deaths as well. And so what's important is to be able to detect early enough. So this is showing a sort of delayed response time. So the normal sending in your test to the lab, you get a response, but now
Mark Cushman.
And so if you take a look at what would happen with a rapid diagnostic type of test, then you're able to create a quicker response and prevent some of the spread of that disease. And so that's what this graph is showing, is how many more cases you can prevent by getting an earlier response. And so that's what point of care type of diagnostics can do, as opposed to waiting a couple of days. You're maybe waiting a couple of minutes or hours to get that result back. And something like this could really be or could have been very beneficial for something like the Diamond Princess cruise ship, where they have almost 4,000 people on the ship. And in order to get testing, you're sending everything off to the lab. And so now you have days where this disease can continue to spread. And so with a rapid type of test, this could have prevented some of those outbreaks or some of those cases. And so point of care testing is exactly kind of what I've been talking about, decreasing the time to that result. And so normally samples collected, samples processed, test is performed. Let me turn on my laser pointer here. And then the test is performed, compile the results, someone has to analyze the results, and now you're finally getting that back to the patient. Whereas if you're able to test at the point of care, everything is done right there near the patient, and you don't have to waste as much time. I turned on the pointer and I don't know how to advance. There we go. So most point of care tests need to follow this assured criteria. So they need to be affordable, sensitive, specific, easy to use, rapid and robust, equipment free, and deliverable. And so all these things are really important, especially when it comes to emergency type of situations, such as a pandemic, or can be very useful in developing countries as well. So currently, the way that COVID-19 is diagnosed is using reverse transcriptase polymerase chain reaction, or RT-PCR. And the way this works is they take a swab, typically from the nose. I haven't seen too much of the throat. It's like about 5 seconds in each nostril. It's not very comfortable, but it's not painful either. And so then what happens is they'll send that off, typically in some type of media, to the lab. Once it's in the lab, they'll extract the RNA from that sample, and then they use the reverse transcriptase to turn that RNA into DNA. And then there are different primers that are placed into the sample that will detect the SARS-CoV-2 DNA. And so after some thermal cycling goes on, if the DNA is present, then it'll fluoresce, and that can be detected using a specialized equipment or a PCR machine. And so again, when we're talking about those point of care tests, one of the things is equipment free. So having this bulky and very expensive piece of equipment is not ideal for testing at the point of care. And so some things that could also happen with this, having to transport the samples to the lab, it could degrade over time, and there's already a low pathogen concentration for these types of samples. And then once you have so much demand, then the personnel and the time can really become a shortage. And so it's really essential to develop more easy to use and rapid tests so that you can limit the transmission and stop the cases from spreading as soon as possible. And so one thing that a lot of groups are looking at for point of care testing is to use a different type of amplification method called LAMP, or loop mediated isothermal amplification. And this one it has its advantages because, as the name implies, it's isothermal, one temperature. And so all you really need to make this portable is a heating source of sorts, and you don't have to worry about the thermal cycling that occurs during PCR. And so another thing that a lot of sources have begun to look at is using saliva as opposed to those nose swabs. And that, of course, would be not painful either. And there have been sources that say that the concentration there is similar to what's found in those nose swabs. So the only difference here between that PCR and LAMP, aside from the temperature, is LAMP is actually a little bit more robust. And so we don't actually need to extract that RNA. So we can put that full sample into our master mix of sorts and then add in those primers that will detect the SARS-CoV-2 DNA. And so that step is the same all the way up to the actual amplification of LAMP. So the way this works is there are 6 different primers. The primers will go and invade that DNA that has been converted using the reverse transcriptase. And it begins to amplify along those strands, and then the product of that forms these dumbbell shapes, and there's more primers that come in and amplify around those dumbbell shapes. So you get many, many, many copies of this DNA. And the way that they can be detected, similar to PCR, we have fluorescent curves. There's also colorimetric detection, where some chemical is placed into the sample. It typically measures the magnesium concentration, and so it will change colors based on that, and that will tell you whether or not the sample is positive. And then I'll talk about our method, which is the particle diffusometry for detecting these LAMP amplicons. So first, we'll take a look at one of the fluorescence methods, which is actually using UV. So to the left here, N15 and O117 are just 2 different targets that they have primers for on SARS-CoV-2. And whenever it's yellow, that means it's positive. So this is actually colorimetric, but this is to give you an idea of what should be fluorescing under the UV. And so, and now that I'm looking at it, it's a little hard to tell. And so this number 1 here has some fluorescence, and then this middle one should also be fluorescing. And so looking at that can be a little bit ambiguous and can be hard to ascertain, but it's a really cool type of method. So this group, they also were doing colorimetric, but in the presence of saliva. And so that was really interesting in our application as well. So with water, without the saliva, their limit of detection was 10 to the 3 for this primer set. And then for the LAMP primer set, they got down to 10 to the 2. And so this is a really good, a pretty good limit of detection for SARS-CoV-2 genomes. And now when you added in saliva, this top one, they're actually more orange. And so yellow is what is positive. And so it's kind of like, is orange positive? Is it half positive? I'm not really sure. Right. So adding in that saliva really inhibited the reaction. And there's so many things in our saliva that could do that. And so then with this LAMP set, they do have actual yellow ones. So we're at about 10 to the 6th here. And so this is not an ideal limit of detection for adding in saliva and being able to actually identify the SARS-CoV-2 gene. So what they looked at was how we could dilute the saliva or different methods to reduce that inhibition. And so without any treatment, as we saw before, there is no change in the color. It's not yellow at all. And so what they did was they added in this heating step, which actually eventually took it up to 95, which is what PCR does. And again, would not be ideal for what we want to do in our application. And I'll explain why, because the things that we add in cannot be taken up to that high of a temperature, and it just wouldn't work very well. And then they added heat and proteinase K, and it's pretty similar to just the heating method here. So overall, this middle one was what they decided to go with, and their limit of detection was 10 to the 2 with saliva in the reaction. So now we can move on to PD-LAMP and how this kind of workflow would go through. And so the patient would take that saliva, spit into a tube of sorts. And so we just saw how the complete saliva does not, it inhibits the reaction. So there need to be some water or buffer in that tube to dilute that saliva down. Then we can take that sample and place it into our chip. In the chip, there's going to be dried down reagents and primers, and along with the fluorescent particles that we need for the particle diffusometry detection. And so what it actually looks like on the smartphone device, both of these videos may be playing. Let me press it. Yep, there. Okay, so these are both playing. It's really hard because they're on a loop, so they're gonna skip and jump. But on the left side is a positive sample, and so this one's gonna have low diffusivity and a high viscosity, right? So that means that these lamp amplicons are actually present, so the SARS-CoV-2 is detected. And what's unique about this is, hopefully you can tell, you can actually see there's some aggregation of the particles here. So what's happening is these green particles are coated with streptavidin. And so we can take advantage of that by adding biotin to one of our primers. And now not only are we changing the viscosity once amplification occurs, but we're also getting a change in the size of the particles, and that we can detect from the diffusion coefficients. So as that particle's Brownian motion changes, we take a video and then we get these diffusion coefficients. And so again, lower diffusion coefficient, positive sample, higher coefficient means that the amplicons did not amplify, the beads are still freely moving, and we won't see a change in the viscosity. Oh, it's stopping the videos. Okay, too far. So this was some of our results from initial amplification just in water. And actually both of these amplified in less than 20 minutes. And what we did was we took, sorry, now the screen is, WebEx screen is in front of me, but anyways. So we went from 7,500 to 10-fold dilutions down to 7.5. So this top one, this is the fluorescent curves. And so it matches the gel, which is pretty much our gold standard just to make sure that amplification has occurred. And so unlike PCR, where you have just strict banding, LAMP actually gives you this smear of sorts and then bands at the bottom. And in the negative control, which in this case was just water, these bands here are just the primers that did not amplify. And so with the second one, we only got to 75 copies per reaction, and that was pretty much representative of most of the repeats. We would at least get to 75, and so that would be our limit of detection in this case. So we're able to detect down to 75 copies per reaction. Our reactions are 20 microliters. This is great, but now we want to really make sure that this reaction is specific to SARS-CoV-2. So what we did was we tested against SARS and Middle Eastern Respiratory Syndrome. And thankfully, the only one that actually popped up was SARS-CoV-2. And so this tells us that even at this high concentration for all of these, it is indeed specific to COVID-19. So that is really exciting. And then we wanted to move on to adding in that saliva. So we took the same LAMP primers, same reaction, and I think initially I actually went up to about 30% saliva, but after 20%, there was no more amplification. And so again, we just have positives and negatives for each of these. Everything's at 10 to the 4 genomic RNA copies per reaction. And 8% and 10% did amplify at this concentration, but as you can see, the bends get kind of lighter here for 8 and 10%. So what we did was we started looking at the 5% to try to optimize that so then we could add in more saliva. So these are the diffusion coefficients that we obtained from our smartphone device. And what's happening here is we have that negative template control again, which is water, and then we have dilutions from 5,000 down. And so for 0% saliva, just the regular reaction, our PD lamp was able to detect down to 50 copies per reaction, which once you divide that 20 microliters is 2.5 copies per microliter. And this is a 30-minute reaction. And so these diffusion coefficients, they're very, it's more so a relative type thing. So it's not actually quantitatively telling you how much of the virus is there. It's still just a yes or no. And so after you do a couple of samples, we can create a cutoff point that after analyzing, the smartphone device can tell you yes or no, the disease is present. And then so when we added in that saliva, we extended the reaction time just to make sure that everything amplifies. So this one is 45 minutes. And again, the same thing, we have 5,000 diluted down to 5 copies. And sorry, I should have put this here. Both of these are N of 4. And so with this one, this is so widespread because there was, I think, 1 out of the 4 that did not amplify the 5E0. And so it was up there towards the negative control. So this is really exciting. We've got 2.5 copies per microliter. And this has all been with just genomic RNA. So what we also wanted to look at was actually using that virus or those virus particles, which still have the genomic RNA. And so with that, we're seeing that same trend. We're able to really get down to 5 copies per reaction, which again is really great. And I'm excited, really excited to move forward doing some more of this testing and diagnostics. So some of the things that we really need to move forward with is trying to up that concentration of saliva. So right now, again, like I said, each reaction is about 20 microliters. So with 5% saliva, that's 1 microliter of saliva. And then the way we do it is we're spiking in 1 microliter of saliva and 1 microliter of the RNA or the virus particles. And so that's pretty small. So it'd be really nice to try to up that concentration. We just have to do a little more optimization. And then we want to do some of that PD lamp on the specificity test. So making sure that there's not a difference when we add in the RNA in the diffusion coefficients, as opposed to just that negative control, which has been water at this point. And of course, we have also been drying down some of these reagents. So another one of those point of care test bullet points is that it is deliverable. And so not having to rely on cold storage or anything of that nature is going to be key in making this a really good point of care test. So we have dried down all the reagents in tubes and on chips, and hopefully looking to integrate all of this soon so that it can be one device altogether. So with that, I'll acknowledge both Linnes lab and Tamara Kinzer-Erson's lab. And this is probably as many of us as will ever get together outside of that. And then, of course, funding and OmniVis source. Oh, I forgot to even mention that. So the phone, I don't know if you saw a couple times, it kept saying OmniVis. And so this is actually a startup company that spun out of the lab a couple of years ago. And it's headed by Dr. Catherine Clayton, who I think was on here at some point too. So that's cool. And then Vodafone for funding. And of course, Weldon School of BME.
Fantastic, Ashlee. Thank you so much for that, and congratulations on great work. It seems like you're making awesome progress and very positive. And congratulations. Yeah, it's great. Can you tell us a little bit more about how this diffusometry technology works? So you said it's related to viscosity, is related to the concentration of RNA. How is the RNA concentration related to viscosity, or as the amplification is occurring, are we increasing viscosity, and so you're seeing the movement of these particles to slow down because they just have more soup to swim in? Is that kind of how it works? Can you give us a little bit more on that?
You just described that perfectly. Exactly. And so when the DNA is actually present, right, it's going to begin to amplify. And so that means there's going to be more and more amplicons in that sample, which is going to make the sample more viscous. And so if there's no SARS-CoV-2 DNA, there's nothing to amplify, the sample's not going to change its viscosity. And so therefore those particles that we've placed there are not going to slow down in a way. Their Brownian motion isn't going to change, and it's not going to have a lower diffusion coefficient than a sample that has more amplicons in it.
Got it. So, and you can do this on the fly in real time, detecting this diffusometry on the phone, or is it, do you have to take time lapses and look at the differences and do that comparison?
Very good question. So we are actually working on a real-time type of detection that will actually see the changes in those diffusion coefficients as the reaction amplifies. But right now it's an endpoint detection. So we amplify the reaction place it in the chip, and then we are seeing what is the diffusion coefficient and what is that change compared to a negative control.
Awesome. Thank you so much. Does anybody else have a question from the audience? We can open it up. We still have a few minutes. And if you want, you can either drop that question in on the chat box, or you can unmute yourself and ask your question. So Ashlee, you mentioned a lot of optimization with the saliva concentration. But what if we were to talk to somebody like Michael Laddish, who specializes in taking and concentrating large volumes? He says he can take a whole lake and he can concentrate it into a 1 ml vial. So if we use, or let's say speak to him, could we improve the methodology for concentrating the saliva without concentrating the impurities in the saliva that might be confounding your PCR reaction, might be inhibiting it or giving you some of this fluctuations that tell you false positives and so on, or I should say false negatives. But can like what? So other than playing around with the concentrations with water, what else could we do to improve that signal and capture more of the RNA in that saliva sample?
Yeah, so ultimately, sample enrichment is something we have also looked at, but at the end of the day, we could at that point just extract the RNA as well, and then we're not going to get, because there are plenty of kits that can extract from the saliva. So that's something we've thought about as well, but of course, in order to reduce the amount of user steps, it'd be ideal to be able to just place that saliva right into our chip.
I agree. I agree. That's really good. Well, thank you so much. If there are no other questions from the audience, I think I had all my questions answered. And thank you, Ashlee, for taking time with us today and talking to us and giving us this awesome update. Let us know how we can help you too in making some connections or whatever you might need. Now it is my pleasure of introducing Caroline Plescia from Rob Stahelin's group. Caroline is modeling SARS-CoV-2 entry and egress using virus-like particles. And so the virus-like particles and virus particles come back. I guess it's a wonderful tool for us to continue to investigate how and how we can detect the virus and how we can chase it around inside the cell. So without further ado, Caroline, please take it away. And I think you're muted, or we can't hear you.
There.
Oh, here we go. Now we got you.
Okay.
Perfect.
All right, so I'm Caroline Plescia. I'm a PhD candidate in the Rob Stahelin group. Formerly, we did a lot of molecular biology research on filoviruses, including Ebola and Marburg. But since the beginning of the pandemic, we've kind of shifted our focus to SARS. So we have lots of people in the lab looking at different aspects of the virus. And my task was to develop virus-like particles, and I'll talk about what those are. So first we have some introduction and background, some development of these virus-like particles that I'm going to use finally to study viral entry and egress. And so that's kind of the overview of what I'm going to be talking about. So COVID-19, we all have heard of coronavirus disease 2019 and the virus that causes it, SARS-CoV-2. I just checked these numbers. Actually, it's very close. The 3.05 million cases in the US, 11.8 million cases worldwide, which jumped quite a few in the last couple of days. That's an approximate 4.5% fatality rate at the current numbers. So obviously this is a huge problem. Not only is this a problem in terms of economics, it's a huge health problem, and we need all of the scientists on it as possible. But currently, to use the live virus, you have to have a BSL-3 or a BSL-4 facility. And what does that mean? Well, BSL is a biosafety level, and it kind of determines the level or the risk of pathogens that you can have in your lab. So BSL-1, which is the most common, works with things like E. coli. BSL-2 is where you find influenza, HIV, and Lyme disease. BSL-3 is where you find tuberculosis, SARS, and other coronaviruses, with high-risk microbes being things like Ebola. And so I mentioned that before we worked with SARS, we did work, or we still do work with Ebola, but we actually run this in a BSL-2 lab. And how do we do that? Well, we use virus-like particles to model these viruses instead of using the live ones. They present a much lower risk. They're unable to replicate inside of you. But as I'll show, they're able to perform many of the same molecular tasks that the live virus is able to. And the reason we want to be able to take SARS down from a BSL-3 and unshackle it and allow the BSL-2 facilities to study it is because in the United States, there are only 13 BSL-4 containment facilities and approximately 1,300. The numbers of BSL-2 labs become exponential with every large major university having hundreds. So that exponentially opens up the research availability if you can take it down into a BSL-2 lab. So I've been talking a lot about these virus-like particles, but first let's talk about what the real virus looks like. So it's a spherical particle that's enveloped with a lipid bilayer that's shown here in gray. It has a single-stranded RNA genome, which is inside, and it has 4 major structural proteins that I'm going to talk about. They're listed here. 3 of them are transmembrane proteins. There's the membrane glycoprotein, the spike protein, and the envelope protein. And inside this lipid bilayer is the genome, which is coded by the nucleoprotein. And that's the last major structural protein. It tethers the RNA to the inside of the viral particle. So that spike protein actually mediates the interaction with your cells as it comes into contact with the cell surface. It interacts with ACE2, which is the cellular surface receptor. This stimulates its uptake by macropinocytosis. It's trafficked into the endocytic system, where it goes through early endosomes and eventually lysosomes. In lysosomes, we know there is very low pH, and that low pH triggers a fusion event between that lipid bilayer around the virus particle and the lipid bilayer that protects the lysosome. This fusion event kind of dumps and pushes out everything that was inside of that viral particle, which would be the encapsulated genome. It's able to uncoat, it's able to replicate, it can produce all of the proteins that it encodes, and it encodes many proteins, but I'm just going to talk about the 4 major structural proteins. And these traffic to what we call the ergic, which is the intermediary complex between, or the intermediary compartment between the ER and the Golgi. So all of these 3 structural proteins will always traffic here. They'll make this nice lattice across the membrane, and then the nucleoprotein is able to meet up with them a little bit later. And this meeting up interaction causes it to bud into the lumen and form a fully formed viral particle, which is then trafficked out of the cell and released into the media. So yeah, we have our 4 major structural proteins that I talked about. So we wanted to be able to model this whole system using virus-like particles and not the full viral particle. So the authentic live virus particle contains all of these 4 structural proteins, its lipid bilayer, and its genome. And we can make these virus-like particles, which look very similar, and they contain everything except that genetic material. So these particles can still self-assemble. They have no genome, but they can enter cells, and they do not replicate, which makes them BSL-2 compatible, as opposed to the authentic live virus, which you would need a BSL-3 to study. So we first started out by just wondering, can we make these virus-like particles with a transient expression system off of plasmids? So we first wanted to study which structural proteins were required for VLP formation. I showed you that all of the viral proteins are present in the virus-like particle, but we wondered, can you still make these particles with a similar viral morphology with maybe just 1 or 2 of the structural proteins, or does it really require all 4? Which structural proteins yield the most efficient VLP production? And can our VLPs that we make be used to functionally model infection? So to produce these VLPs, we use a transient expression system. Here I'm showing co-expression of the 4 major structural proteins at once. So regardless of whether they're expressed off of the viral genome or whether they're expressed off of plasmids, these proteins will still be expressed in the cell and they will traffic to the same place. And that trafficking to the same place allows them to still self-assemble into these hollow, empty viral particles that are still released from the cell in the same way. So about 72 hours after transfection, I can collect the media off of my cells and have these viral particles just floating around in them. How do we get them out of the media? Well, we load them onto a sucrose cushion, do ultracentrifugation, and we get a nice concentrated pellet. These are pictures of pellets that I've gotten. So you just resuspend that pellet in whatever buffer your virus is suitable in, and you have your solution. So before we jumped into doing this with all 4 of the structural proteins, I wanted to assess the ability of each one to make VLPs itself or possibly be secreted. So I did a VLP collection from the media when expressing only 1 structural protein. I also collected the cell lysates and did Western blot analysis on these to show whether the viral proteins were present in the cell and VLP fractions. And I'm going to show you some Western blots that have some slightly confounding results, and I'm still working on them. But if anybody has suggestions, I will take them. So here in our mock sample with all of the negatives, we don't see any protein but our loading control. When I express M alone, interestingly, we do not see it show up in the cell lysate. I'm going to talk more about this, but we think that there's some kind of cellular degradation going on when this protein is expressed alone. When co-expressed with other proteins, it doesn't show up in this blot, but I'll show you other blots later where it does seem to be stabilized and protected from some kind of cellular degradation, so it will show up later. The nucleoprotein expresses quite well in both its alone and co-expression conditions, and in a similar scenario as M, we see that when E is expressed alone, we see very low levels, indicating that it's possibly targeted for some kind of cellular degradation. But when it's co-expressed with other viral proteins, they seem to be protecting it in some manner. Finally, when the spike protein is expressed alone and in combination, we see it show up quite well. So this is what is being expressed in the cells. Next is what we see in our VLP fraction. So again, when M is expressed alone, we see no M in the VLP fraction, but altogether we do see it show up, indicating that when all 4 of the proteins are co-expressed, they are able to form these VLPs, which exit the cell and end up in our VLP collection. We did see a little bit of N show up when expressed alone, and this is either a novel ability of SARS-CoV-2 to produce VLPs or be secreted, but it's not reported for any other coronavirus. So this could just be an artifact of our overexpression system. Finally, when co-expressed, we see it show up very bright in the VLP fraction. Again, we don't see E released, but when co-expressed, we do. And again, with S, which is hard to see in this blot, you don't see it alone, but you do see it when it is co-expressed with all the other viral proteins. So this was assessing each of the structural proteins alone. Then we wondered, because M is so important in maintaining the morphology structure of the virion, would different combinations of these plasmids with M induce VLP formation? So here I'm showing the same type of blot, but we have different combinations of M with N, E, and actually I'm going to do combinations with M and S as well. I just haven't quite gotten to it yet. But I do show that in the cell lysate, I can now get M to show up, but it still doesn't show up when it's expressed alone. So this does indicate to us that maybe there is some kind of cellular degradation going on, but co-expression with these other viral proteins protects it from whatever that pathway is. So in the VLP fraction, Again, we don't see M show up when expressed alone, but when you co-express with N, it shows up quite nicely, indicating that M and N together are the minimal, or one of the minimal systems for VLP production. And you see this increase in the intensity of M, which indicates to us that as you add in more viral structural proteins, you're getting a more efficient assembly and release of these virus-like particles, which makes sense. As the system becomes more realistic, you're going to get a more realistic output. So the more structural proteins that we provide, the better it's going to work. And over here I have a quantification of the release of viral particles, and we see the most efficient release with M, N, E, and S, as I just said. But we do see some VLPs with M and E, but it seems like N is really the driving force here. So we wanted to know if the release of these virus particles was going to be a good model for cells that would be releasing the live virus. Oops, I meant to talk about these, but you see the same patterns with N and E and S. The bands get more intense as you include more viral proteins into the transfection. So I'm sorry, before I talk about the release, we wanted to assess the structural integrity of these virus-like particles and make sure that we weren't just co-sedimenting secreted proteins here. So we did transmission electron microscopy on our mock samples on a VLP collection from cells co-expressing M, N, and E. And here you see this bald particle that's about 100 nanometers in size. But as you add in that spike protein, which is known to give these particles their pronounced crown or corona, you do see that in the micrographs, and you can see that here. And we thought these looked spectacularly like the live virus in this high resolution photo that was published by the CDC. We can see the spike protein, maybe not to the same resolution, but it is definitely being incorporated into what we are now calling virus-like particles. So we wanted to know if the release of these structurally intact particles was going to be comparable to the live virus. I said that these particles are released by exocytosis, but in live virus infected cells, we see these long filopodia that seem to come out of the cell and carry the viral particles away from the cell that produced them. This is a mechanism by which virus is used to spread, and we wanted to know if that would be modelable by our plasmid-based system. So we did scanning electron microscopy, again, of different combinations of M protein with the other structural proteins. When we did mock transfection, this is just a wide field image of what you see on the electron microscope. These are each individual hex cells. You can see that most of them are pretty smooth, but some of them do have a little bit of filopodia. This is a close-up picture of what that looks like. Again, it's mostly smooth, but it's got some filopodia protrusions. So when we co-express M and flag E, we do see the production of some VLPs. However, they seem to sit on the surface of cells. They don't seem to be carried away by these long filopodia. But when we co-express with N, we start to see these formation of these long filopodia that are carrying an abundance of virus-like particles. And we see the same thing when we include the S protein. So altogether, we think that our VLPs and the release of them is a good model for the live virus because we can even induce the formation of filopodia with our plasmid-based system. So we've shown that the egress is modelable, but what about entry? So these virus-like particles, I needed a way to track them as I put them on cells that I wanted to infect. So what I did was I made a GFP tagged version of these VLPs by incorporating a GFP tag onto the spike protein and co-expressing that with the other structural proteins. Here's a Western blot that just shows the incorporation of the GFP tagged S protein and that I can get it in the VLP collection. But I wanted to take these GFP tagged VLPs and infect cells. So what you do is you just take this infectious media with the VLPs floating around, you put them on your target cells and spinoculate. That's when you put the cells into the rotor, you spin them at 4 degrees for about an hour, and it primes the viral particle onto the cell surface receptor so that when you take it out of the rotor and you put it into the incubator at 37 degrees, you can stimulate the immediate uptake of the viral particles. And we let them incubate for about 2 hours. This gives them just enough time to be internalized and trafficked into the endocytic system. And then we stop that reaction with fixation and staining for confocal imaging. So when I took pictures of these cells that I infected, first I did a mock infection. What you're seeing here, the blue is the nuclei, the red is the plasma membrane. The binding of this dye binds to sialic acid, so sometimes it's internalized by endocytosis, which is why you see some little puncta. And the green channel will be our GFP VLPs. So this is the mock infected. When I did a real infection, we get these really, really nice bright green puncta, which are concentrated GFP VLPs inside of different vesicles internalized into the cell. And to just highlight how many there are, really, I took Z-stacks through the cells, and you can see there's just an abundance of these VLP positive vesicles. Finally, I wanted to know, am I seeing VLPs in the correct vesicles? Correct being comparable to the live virus. Is it entering by the same mechanism? I wanted to know how good my model was. So the live virus is known to be trafficked through early endosomes, which are Rab5 positive, and lysosomes, which are LAMP1 positive, but they're not known to ever enter these vesicles, which are called peroxisomes, and they are PTS1 positive. So these are the three markers I used to do co-localization analysis with my GFP VLPs and to see whether they were entering these specific vesicles. So when I mock infected my target cells, I'd see my nuclei, but I don't have GFP signal or M-cherry signal because I used an M-cherry marker for each of the vesicles. When I used M-cherry Rab5, that marker for early endosomes, you can see co-localization denoted by the yellow appearance of the green dots in this merged image. That indicated to us that there was co-localization with early endosomes as expected. With LAMP1, which is that lysosomal marker, we have co-localization again. But when you infect cells that are expressing the peroxisome marker, these look quite green, indicating they are not co-localized with the peroxisomes. And so we've quantified that here. It's in the very last panel where our mock has a non-significant co-localization, but there is significant co-localization with M-cherry Rab5, M-cherry LAMP1, but not PTS1, indicating that our VLPs are utilizing the same entry system as the live virus. As controls, I expressed GFPS alone and collected the media off of those cells and used those to infect my target cells just to make sure that GFPS alone wasn't being secreted and entering cells. And I saw a non-significant co-localization because there wasn't really any green signal in any of the images I took for those. So this is really where we're at. We've done all of this in the last 2 or 3 months. We've been able to show that the SARS-CoV-2 VLPs are self-assembling. We've shown the minimal system for production. We've shown that our VLPs are released realistically from cells on Philopodia, that the VLPs can be fluorescently tagged, and that our fluorescently tagged VLPs can be used to study viral entry. And so just some acknowledgements. The Stahelin lab, which is pictured here, my PI, Robert Stahelin, our lab manager, Nathan Dissinger, Emily David, who's a third-year grad student, and Dr. Patro, who is a postdoc. They did a lot of the electron microscopy imaging. And I also like to thank the Purdue Pharmacy Live Cell Imaging Facility, the Purdue Electron Microscopy Facility, and my very supportive friends and parents. So I can take any questions you have.
Awesome. Well, thank you so much, Caroline. That was great. And congratulations to you on all your progress and great work that you're doing. This is fascinating. I'm not sure if anybody else in the audience has questions. Mark, I see you unmuted yourself if you have a question.
Yeah, I have a question. Hi, Caroline. It's Mark Cushman.
Hello.
I have 2 questions. One, do you think your VLPs have any potential use as a vaccine? And the second question is, could you use your VLPs to study compounds that inhibit the entry of the virus, maybe like in a high throughput screening format? So thank you.
Yeah, the answer to both of those is yes. They definitely could be used for vaccine development, and I think there are people trying to do that currently. there has been talk of using our purified VLPs and giving them to labs that want to do that. So we may be participating. And currently, we're actually trying to get, basically, I want this to be readable by a plate reader and not readable by confocal so that we can do drug screening. So we're working on optimizing that as a plate assay. It's been a little finicky, but I think it's starting to work.
And yeah, me too, Mark. I'm thumbs up about both of those answers. Very exciting. I would also recommend that you may want to get in touch with uh, oh, oh my goodness, the drug discovery chemical genomics facility, Lan Chen, because they have a PerkinElmer Opera Phoenix with a confocal in it to be able to do live cell monitoring for this type of screening where you have to do image analysis and you can automate that image analysis. So I appreciate that you're trying to simplify it to take it to a spectrophotometer or some type of, automated plate reader type of screening, but you could start or you could do some great progress with using that Opera Phoenix that Lan has in her lab. So that's another way of doing and maybe bringing what the ideas that Mark has behind his questions to fruition in a type of setting like that. Caroline, fantastic. My question that I had was, I'm really interested in now. You sparked an interest in me about the dynamics of the timing of assembly. Do we know any idea of what comes first and who comes second, and like what's happening in terms of the assembly timing and how things are made? It seemed like you had cooperative type of interactions between these structural proteins, and what does that look like? I mean, if it were me, I would try all the algorithms, all the combinations, but that's just me. Obviously, we have limited time and limited hands. So what are your perspectives there, and how are you going to try to decipher that if that's what you're going after?
Um, well, we have an idea of how it works based on SARS-1. I don't know specifically about the timing, but I know the M protein oligomerizes across the ergic membrane, and it's able to then incorporate the other two integral membrane proteins into that lattice, E and spike protein. Um, the nucleoprotein in the live virus infection would probably only get there once it was bound to like genomic copies. So you'd have to wait until it was in the replication, end part of the cycle to carry those genome copies to the ergic and meet up with the other viral proteins. But we get, after 72 hours of incubation with the plasmids, it's obviously happening in under 72 hours. I can detect them at 48, but not really at 24. So there does seem to be some type of lag time there. And I would assume that might be with the nucleoprotein meeting up at that membrane because it usually wouldn't go there until it had the genome attached. In terms of looking at the different combinations, yeah, I would really like to look at all of the different combinations, especially with N, because we see it's like secreted or something, doing something weird. And that is definitely our next step.
Totally. It just seemed really like you're piecing together some of that puzzle, and it just seemed fascinating. You captured really my attention in that piece of the presentation. I love the images, and I really like where you're going with that. Are there any other questions from the audience? I wanted to ask you, Caroline, is it when you were talking about the Filipodia extensions and how you were kind of taking those images for those, was there is there anything to say about ACE expression in the Filipodia and in those particular cells as being somewhat hyper expressed in those areas with the Filipodia and trying to pull out how how's the dynamics there working between the virus and ACE expression, ACE receptor expression?
So I don't think anyone has looked at that quite yet. The paper that shows the Filopodia and the live virus infection just got released maybe a week ago, 2 weeks ago. So that's really new information. I know in that one, they talk about the Filopodia being actin-rich and being speckled with viral proteins such as N, but they didn't say anything about ACE2, and I think it's really hard to guess whether there will be ACE2 there or not. But that would be an easy experiment to do with confocal to see if ACE2 was on Filopodia that we saw extending from plasma membrane.
Right. That's great. And so what's next, Caroline? What's the next step for you other than, okay, let's create the assay for improved throughput and scale so that we could start doing some drug discovery. There's that route. Are you guys also pursuing the vaccine route and looking at how some of these virus-like particles could be used as vaccine particles?
I know that my personal interests lie more with the drug screening and drug discovery over the vaccine. But I think it would be silly of us to not contribute to the vaccine development as well. I think we should give our viral particles to a lab that knows what they're doing. Also, somebody just sent me a question. Let me read it. So how difficult is it to produce and purify the VLPs? Could other researchers use them too, easily in cell culture system or in labs? Yeah, they're time consuming and a little cumbersome, but they're really not that hard to produce. It just takes time to load that media onto the sucrose cushion and to spin it, because we spin it for about 3 hours. But other than that, all you have to do is the transfection and collection, and then the Western blots honestly take the most time. But if you want to collaborate, you could always just talk to me and I can give you purified VLPs. That might be much easier.
That's great. And thank you so much for that generous offer. I also want to remind the people that are joining us today that this is also a way to exchange ideas, exchange information, and also create partnerships like the one that Caroline is describing here, where we're trying to put together this series so that we can highlight the great work that is going on, but also identify potential opportunities for partnerships and collaboration. So if at any point in time you're seeking reagents or you need particular collaborators or resources, please send us an email, send me an email if you want, and would be happy to connect you. Are there any other questions, Caroline or Ashlee? Anybody receiving anything on your end? And if not, I really want to thank you both for giving us great summaries today and fantastic presentations. Congratulations on all your progress, and we're looking forward to continue hearing the great work that you're doing and seeing some of these in publications and really hot pursuit of innovations and technologies. And with that, I want to thank everybody else for joining us. We are going to be doing this again in 2 weeks' time. I still don't have my speakers lined up, but look out for your emails and look out for our newsletters where we will be announcing the next set of speakers. And again, to remind you, this will be on July 22nd, same bat channel, same bat hour. And I hope you take care, be safe, and thank you for joining us this morning.
Thank you.
Thanks. Bye-bye.